Good morning if you're here to join me. What we've got today is a more polished version of the practice live stream I did on Friday. So what I've got is, if you've seen the post last night, you would have seen the toolkit with all the components that you need for this. Now, if you've got a tablet with a stylus, when I say stylus, this is all I use. I think it was like £17 off Amazon, okay? Click it on, the blue button comes on, and automatically it pairs and you can drag and drop. So how I do this is, all I simply do is I'm using Notability, I'm clicking on the lasso, okay, but it comes in the form of a box, I'm not doing it free form, and I'm just going to highlight it, and then I'm just going to move all these bits, and what I'm going to do is piece together the Calvin cycle. So it should look something like this when it's finished, to keep it relatively simple and straightforward. So, to put it into context, right, the first thing that you need to know is that the Calvin cycle in photosynthesis takes place in the stroma, which is the sort of space in the chloroplast, not the green sort of stacked coins that contain the chlorophyll pigment. So it takes place in that there. So if you're writing an essay, that's a fact you automatically must put in. Looking at this, see that it's a cycle and arrows pointing the way that things progress through the cycle, or as that input into the cycle. So that's another thing that you need to know. It's a cycle, it's powered by energy, right? And you maybe see straight off the bat, there's energy there, there's ATP, right? So it's also powered by enzymes, right? So straight away, it's a cycle, it takes place in the stroma of the chloroplast, and it's a series of enzyme controlled steps, because we're going to change from one thing into another, and if we change something into another thing, we're going to need an input to be able to facilitate that change. Okay, so two more things that you need to be aware of. In photolysis, the first part of photosynthesis, we create NADPH and ATP, which are going to be produced in photolysis to move on to the second phase, the Calvin cycle, right? A couple of notes. Phosphorylation, ADP plus PI, brings about the high energy compound ATP, and the hydrogen acceptor NADP will pick up hydrogen and be converted into NADPH, the reduced hydrogen acceptor. Reduced hydrogen acceptor means it sort of loses its ability to gather any more hydrogen onto it. So that's the background info for it. So let's go on and we'll have a look at actually making this diagram. Okay, so with all the tools that I've got, let's ignore that screen time notification. With all the tools I've got, I'm just simply going to drag and drop. If you can do this, right, if you can make all these sort of bits and pieces, then do it in your own time, or rewatch the live video, and then you can start to piece it piece by piece in your own time. Feel free to pause, start again, but just make sure you get it right. There is a few sort of complicated parts, but we'll try and eradicate that. So it's always good to start with a title. So we'll go with, so I'm just going to take my lasso, go with the title, and I'm just going to bring it onto a new sheet, which gives us the Calvin cycle. Okay, so the Calvin cycle, as it suggests, as we've looked at previously, it is indeed a cycle. So we're then going to bring in the next part. Okay, bring in the next part, and that's going to give us the wheel that gives us the cycle. Where do we start? Now what I'm going to do is I'm going to begin with carbon dioxide, right? The Calvin cycle begins with carbon dioxide. So I'm just going to simply drag this down into place. And what we're going to do is we're going to get that CO2 into the cycle. We're going to feed it into the cycle and fix it with something, right? So there it is, nearby in the cycle. And I'm going to take this first arrow, okay? and that first arrow is going to feed into the cycle like so. Now, when the CO2 enters this cycle, there needs to be something waiting for it to be fixed towards, because at this point in time there's nothing. Okay, as I'm going through this, there's nothing. So the CO2 enters the cycle and it's going to meet this compound here, which is RUBP, and the CO2 is going to meet it. So the cycle is going to be turning. The cycle is going to be turning. I'm just going to change the colour on that to black. It's going to go that way to indicate that the RUBP is going to meet the carbon dioxide. So RUBP and CO2, when they come into contact with each other in higher biology, nothing happens. Okay, nothing happens at this stage. However, we need to get them to join together. So the thing that's going to join them together is the enzyme. And in this case, the enzyme is going to be Ribisco. So the Ribisco is going to take place right about here. Okay, and we're going to feed that into the cycle. Okay, Ribisco in the cycle. Let's make that a wee bit more. Yeah, perfect. Okay, so by the enzyme action of the Ribisco, RUBP is then going to join with the CO2 via Ribisco. So RUBP and Ribisco is going to fix the CO2 to it. Now, when you add something to something, you create something different. It's not the same thing anymore. Okay, so that's key thing. So if you're going to change something, in this case you're going to change the RUBP, adding CO2 to it, fixing it with Ribisco, you're going to create something completely different. So in this case, what we're going to create is 3PG. So if we take this one, okay, we get the 3PG and put it in an equation, RUBP plus CO2, and then if you put the enzyme Ribisco over the top of it, you will end up with this organic compound 3PG. 3PG at this stage still isn't what we're trying to create, because ultimately what we're trying to create here is a sugar molecule, glucose, to do amazing things with it. So 3PG, we need to make another change from it, and this point here is where we bring the products from photolysis in the first stage, the light dependent stage. We bring it to the carbon cycle and we input them. So they are going to be the NADPH. Okay, I'll take that down. NADPH, and it's going to be put into the cycle. Okay. Oops. I've made that a bit big. But there we go. NADPH into the cycle, so that's the first thing. Now, what happens here is the hydrogen gets input into the cycle. And this sort of makes sense because if we're trying to make a sugar, okay, carbohydrate's fancy name for sugar, and if we split it into elements, you get carbon, hydrogen, and oxygen. Okay, so we've got the C, okay, we've got the O, and now the NADPH, hydrogen drops off from it, and that gives us the CHO that's going to allow us to create the sugar. So that's the first thing taken care of. Once the hydrogen leaves the hydrogen acceptor NADP, it's then free to go and collect more from photolysis again to keep the cycle going, to keep the supply chain of ingredients going on for it. Right? Now the other thing that was produced is, tap that, is ATP. Okay, ATP gives energy. So alongside the hydrogen, ATP is then input into the cycle, and with the addition of the hydrogen, and the high energy compound ATP, the 3PG is now going to be changed into the organic compound G3P. You'll see that they are very, very similar, 3PG and G3P, right? Very, very similar, which can be confusing, but how I remember it is alphabetical order, i.e. numbers, sorry, not alphabetical order, numbers before letters, and then PG is first. And I remember PG because it's how movies are rated in this country. So 3PG, and then the second one after the hydrogen and the ATP's been input, convert into G3P. So we've now got G3P, and you'll see in the cycle that G3P, its next stop looks like it's going to be RUBP at this point, okay? But what then happens to the G3P is we're going to create that desired product, the glucose, okay? But we also cannot exhaust that G3P, because if we did, then the cycle would stop, okay? So what we'll do to the G3P is we'll take some of the CHO out of it and take it as a product glucose, but we'll have some remaining, and basically G3P minus glucose leaves you back with the RUBP, which allows the cycle to continue. If we used all of the CHO completely, we wouldn't have the RUBP left to go on and do it again and again and again, because plants constantly need to create food. So it wouldn't make sense for it to totally exhaust its means to create that food. Okay, so that then brings us to the next part. We've got, I'll just bring this down here. So from this, okay, so from that we create glucose, okay, and glucose it's like a preferred substrate for the plant's metabolism, day-to-day living, etc. It does basically everything, but you can't use all that glucose because there needs to be, if you can imagine, other useful uses of the sugar, right? And this takes you all the way back to first year, okay, when we look at a basic plant cell and a basic animal cell. And you'll know that a main difference between them is the cell wall. From National 5, you should know that, in fact you probably know this from first year, you should know the cell wall is composed of cellulose. So that glucose that we make can have 3 different fates, okay? So the first one we just spoke about just now is going to be cellulose. And when the plant when the plant starts to grow, it undergoes cell division. There must be cellulose present to create those cell walls composed of cellulose. So we get that from the second stage, the carbon cycle and photosynthesis. We also get glucose, which is used immediately by the plant for its day-to-day means, mitosis, etc. It's preferred food source at this point in time, so things like mitosis and things like that, it's going to be used for that. And then finally we've got the storage sugar, okay, which is really used for a rainy day. Okay, so the plant can store that starch, and in times where, maybe the winter time, when there's not a lot of sunshine and the plant wants to still keep producing food, it can then call upon its starch reserves to break it down into glucose and then create, it could be more cellulose or it could be glucose for day-to-day things. So in a nutshell, that is the carbon cycle, where we go and we've created the whole thing that you need to know higher biology in Scotland. and we've created this exactly like for like, following the steps that I've took you through. So if you want, have a go, and if any of you do it and come up with this carving cycle that you've done on your own, then post a picture into the community thread. And if there's any questions on it at all, anything you think I've missed out on, because I'm not perfect, I'm not a genius by any stretch, but I'll try my best to help you in case there's anything that I've missed in this. Okay. Now tomorrow morning, I'm sure tomorrow morning in my post, I'm either going to be, yeah, it's going to be genetic engineering 1.5, and I'll do the exact same thing. Okay, so that's aimed at National 5, and then on Wednesday I'll do Advanced Higher, where I'll look at nerve impulse transmission. Okay, right, I hope to see you soon. If you like this, feel free to share it to anybody, and also please subscribe to the channel. That would be a great big help, and I'll keep doing this for you. Okay.